Hello, my name is Pearl Huey. I'm a biomedical scientist working in the cellular pathology department in Betsy Cadwallader University Health Board. The background to this project was a passionate plea from our clinical colleagues in respiratory medicine to try and help them improve outcomes for our lung cancer patients. This project brought together multidisciplinary teams from across Wales to evaluate the possibility of using a fully automated platform to deliver rapid EGFR molecular testing locally. This is in order to help optimize treatment options for very ill patients who present with late-stage lung cancer. These patients may currently miss out on the opportunity to benefit from life-prolonging targeted therapy, as they are often too ill to wait for a full molecular profile from Cardiff before initiating treatment. Due to COVID-19, patients are delaying seeking medical advice, and hence are likely to present with more advanced disease requiring urgent medical intervention. Non-small cell lung cancer accounts for around 85% of lung cancers in the UK. Depending on the patient's medical status and stage of disease at the presentation, treatment options include surgery, radiotherapy, chemotherapy, and more recently, personalised targeted medicine. Identifying specific genetic mutations associated with this type of lung cancer can help determine which patients are more likely to benefit from one of the many new targeted therapies. In the UK, NICE recommends that PD-L1, ALK, ROS1 and EGFR molecular testing is routinely carried out for all adults with previously untreated, local advanced or metastatic non-small cell lung cancer. This is in order to inform clinical management. Around 16% of these patients will have a mutation in the EGFR gene, and these patients will show a greater response to EGFR tyrosine kinase inhibitors than they do to traditional chemotherapies. Conversely, patients without such mutations respond better to conventional treatments. Clinicians therefore need to be able to identify each group of patients to ensure optimal treatment. Molecular testing can be delivered in a variety of ways. Immunohistochemistry is a reliable method for PD-L1, ALK and ROS1 rearrangements, but DNA sequencing methods are recommended to test for EGFR mutations. Next-generation sequencing is the method of choice, as it can screen a large number of cases for a wide variety of mutations at the same time. However, most genomic testing methods involve complex preparation of the specimen in a clinically clean environment. It also requires specialist training of laboratory staff and extensive clinical expertise to ensure correct interpretation of results. This often results in a turnaround time of around 2 weeks. It is important at this stage to highlight that NGS is widely regarded as the gold standard for molecular testing, and use of the rapid near-patient tests are only intended as an adjunct to be used only when deemed clinically necessary. Lung cancer is the most common cause of death from cancer in the UK. Between 2016 and 2018, there were over 6,500 lung cancers diagnosed in Wales. Many lung cancers are not detected until they reach stage 4, when the cancer has already spread. These patients are often very ill, and in many cases the results of molecular tests are needed the same day. This is in order to decide on the most beneficial therapy. Unfortunately, Welsh patients currently have the lowest overall survival rates compared with other UK nations, and Wales ranks 27th out of 28 European nations. Time to treatment translates into survival, as seen here in the Cancer Research survival statistics. To arrive at a diagnosis, a small sample of tumour is removed, processed in the local histology laboratory, and the sections produced is viewed by a consultant histopathologist to deliver a diagnosis. In an attempt to expedite urgent reports, most histology labs have already implemented local testing for PD-L1, ALK and ROS1 using immunohistochemistry techniques. But this is still only of limited benefit, as the oncologist also requires EGFR tests to initiate treatment. EGFR tests are currently sent to the All Wales Molecular Genetics Lab in Cardiff for DNA sequencing using next-generation sequencing equipment. Specimen transport issues and the complexity of testing results in a turnaround time of around 2 to 3 weeks for most molecular tests. NICE guidelines, the optimal lung cancer pathway and the suspected cancer pathway all recommend a targeted turnaround time of no more than 3 days for histology, plus an additional 10 calendar days for molecular testing. This project had a long list of tasks. Firstly, to evaluate the anecdotal need for urgent EGFR testing. We audited the turnaround time to identify all the pinch points in the pathway. Next, we investigated what fully automated platforms were available for rapid EGFR testing. We then validated the platform identified, developed a standard operating procedure, along with defining a user guide. Finally, we spent some time working on speeding up the transmission of reports. The user survey shows a clear requirement for a rapid EGFR test for a limited group of patients. 100% of respondents saying they would like a quicker result. 3/4 of those questions had encountered patients who would have benefited from a rapid EGFR result. An independent review of available testing platforms was undertaken. It was specified that we required a fully automated platform that required no specialist premises. The platform had to be CE approved and well established in other reputable labs within the UK. It had to be easy to use and provide clear, easily interpreted results with built-in quality control. We needed minimal manual intervention so as not to impact on the workload of already busy histology laboratories. The system had to be cost-effective and have the ability to deliver a reliable, clinically actionable result within 24 hours of diagnosis. The study confirmed that the iDILA was the only platform currently available to meet all these criteria. The iDILA platform is compact and no special clean environment is required, very simple to use with no extensive training required. The results are available within 3 hours of requesting the test. The cassette contains a panel of all currently clinically actionable mutations in exons 18 to 21 of the EGFR gene. The system is also CE approved and currently in use in over 50 labs in the UK. For the validation, 93 tissue samples from patients with non-small cell lung cancer were examined. 97% of cases showed concordance between the NGS and the iDILA results. 74% were negative by both methods, and 23% were positive by both methods. However, 3 of the results were discrepant. All 3 were very rare mutations detected by NGS. None of them are currently clinically actionable and hence were not included in the iDILA screening platform. A standard operating procedure was prepared, mapping out the procedure and detailing the limitations of the technology. The SOP also detailed essential quality control scheme enrolment, as well as training and qualifications required to deliver the report. Whilst the All Wales Molecular Genetics team did not wish to endorse the iDILA platform, they kindly agreed to provide advice to assist in its implementation in histology labs. This is in order to facilitate a rapid result when the clinician deemed an urgent report was needed to direct patient care. In order to alleviate concerns raised by the genetics team relating to reliability of the iDILA system and the quality of interpretational comments, a number of reporting boundaries were agreed, namely that local testing would only report on 2 common mutations. The good news is that 85% of patients with an EGFR mutation are likely to have one of the 2 common mutations, indicating that they are highly likely to respond to tyrosine kinase therapy. This will be a real benefit for the majority of very sick patients needing an urgent treatment decision. So what are the benefits of tyrosine kinase treatment over traditional chemotherapy? Tyrosine kinase inhibitors are given orally. This in itself has many benefits. TKIs are less toxic, they improve the quality of life for the patient, they delay the need for chemotherapy, and they also can significantly extend patients' life expectancy. It's also worth noting that many non-small lung cell cancer patients are under the age of 50. Many have young families and dependents. Increasing longevity and improving quality of their life has a very positive impact, not just on the patient themselves, but on the whole family. How does this project deliver the Bevan principles? Well, what this development does is to ensure the needs of the patient are at the heart of lung cancer care. Right test, right time, right place. Keeping patients out of hospital for as long as possible is a very good example of prudent healthcare. In conclusion, testing for EGFR mutations in very sick patients with stage 4 non-small cell lung cancer can safely and economically be delivered locally within less than 24 hours of diagnosis using the iDILA platform. This can be done in a routine histology laboratory, enabling patients to have the opportunities to benefit from tyrosine kinase inhibitors therapy where appropriate. This improves and extends the quality of life for this group of patients. The next steps, once funding is agreed, we are ready to go. We will need to enrol in the required quality control schemes. We will need to communicate with clinicians on the new testing pathway, and it will be great to continue to build on the work of this project in developing a hub and spoke model for molecular testing. The Bevan project for me has been an excellent experience, providing new skills and opportunities to play a part in improving patient care. I would personally very much like to thank the Bevan Commission team, Prof Rich, Mr Adrian Thomas, Sean Charles and Mrs Lauren Fear for the time they have invested and the personal encouragement I have received from them on this journey. Thank you. Diolch yn fawr iawn.
We will now have a Q&A discussion. Is the platform available at a national level? And if so, why did you decide to bring it to a local level?
Yeah, the platform's a commonly used platform. It's in use in over 50 different laboratories across the UK. However, Wales haven't adopted it. Wales have a strategic sort of pathway for molecular testing. But what we're finding is that it's very directed at getting the full profile of the patient, which is great, and it's great for research and for other things as we go forward. But what it's not delivering at the moment is it's not allowing the clinicians to treat the very, very sick patients that they're seeing coming in who need treatment the same day. The best we can get from Cardiff Molecular System is a 14-day turnaround. So we're just on that at the moment. We're very lucky in that our turnaround time is probably better than most other labs in Wales. So we are directing that, but having this platform would allow us to actually do the test locally, get the result to the patient, not to the patient, to the clinician within a matter of hours once a non-small cell lung cancer has been diagnosed. I think one interesting thing was that a lot of our patients go to Liverpool, and if we were to send our tests to Liverpool to be tested, they'd be testing them using the Idilla system and giving us the results within the time frame that we'd need. So it's been how shall I say, a challenging project to try and change the pathway that's in place, because there's a lung cell cancer pathway and that's in place, and what you do is steps in that pathway, but perhaps varying from that pathway needs to be something that we need to do for certain patients, and it's almost like a one size doesn't fit all method. And this is what we've been trying to highlight by bringing this project up, is that we need more of a patient focus rather than a technology focus. We can't be driven by the technology. We've got to be driven by what the patient needs.
What barriers did you face and how did you overcome them?
Yeah, we faced quite a few barriers. I think the barriers is generally this is the way that we do things in Wales, and I think trying to veer from that has been quite challenging. So what we did do is we asked the molecular service to come on board with us. They very kindly went through and helped us a lot in when we were developing. They were very critical friends, shall we say. They pointed out a lot of the pitfalls, which was very useful because we probably wouldn't have known them otherwise. But what that did was enable us to put actions in to mitigate any of those risks and pitfalls within our new proposed testing procedure. So the testing procedure is pretty watertight now, I think. They've also highlighted what the training they could help us with to move forward, and what we need to go forward to develop perhaps a hub and spoke system with perhaps a possibly next generation testing system up in North Wales, or perhaps another platform that we could join in. But it's trying to build the links and develop those links. And I think people are probably the most important thing in trying to build these changes into place. And we've had a whole team in North Wales and Swansea. And I think between the two teams working together, we've been able to evidence the sort of accuracy and the efficiency of the Idilla platform, and sort of highlight why it would benefit. It's not a huge number of patients, but it's a significant number of patients that would actually have tyrosine kinase treatment, which they may not have had previously.
Have you got a strategy for disseminating your findings further afield?
we're not quite sure how we roll this out, to be honest. I think the next step is understanding how how we can actually roll it out in North Wales and in Swansea to start with, and then make sure it's working properly, and then bring other health boards in. There's only 6 health boards in Wales that do histology. And with all 6 health boards having this advantage, it would provide a huge benefit, but I'm not sure of the support we'd get to roll that out. So we're still in we need the support, we need um, the local health boards to decide that they need to go forward with this. And it might not be directly in the pathway of the cancer services sort of strategy for molecular testing, but it's something that perhaps people weren't aware of in Wales because it hasn't been publicised, and trying to raise that publicity. It's been done in a lot of the other UK nations. They're performing better than us. It's been done in a lot of the rest of the world. They're performing better than us, and we need to do something to improve. So I think we've only got a small group of histologists. They're all aware of the project. They're aware of what we've been doing. And it's just trying to step over that barrier in a way to move it forward, and sort of get people to believe in what we're doing and share that belief and sort of support it. What would be really nice if we had sort of support from Cardiff to help with the interpretation. Sometimes we're very limited in what we can report at the moment, but if we had more knowledge and support around what we could report as we go on, I'm sure we'll be able to report more. And what's really interesting is that the Idilla platform is now bringing out a new cartridge, which will test for all the molecular markers. So that's associated with non-small cell lung cancer. So it'll test for a whole range, whereas now what we're doing is only looking at EGFR, and then we're doing ALK and ROS1 and PD-L1 by immunohistochemistry, which is great. But the EGFR marker would be included in the cartridge with NTRK, RET, MET, and ALK and ROS1. So there'll be 5 or 6 different mutations we could find in there. So the next step may be to validate that sort of cartridge and move that on, so that we get a wider screen for the whole of the lung cancer and not have to send anything to Cardiff for the molecular on these very sick patients.
What advice would you give to future Bevan exemplars?
I'd say if you've got an idea, then share it. See what the sort of the need is for it. We've shared this with our lung team. That's the lung team that came to us, and we've built it from there. The support from the Bevan Commission has been fantastic. It's been really, really good. Lots of advice, lots of training, and just pulling the team together. There's a lot of the team dialed in today, and I think the links we've built up, and the way we've looked at how we can overcome obstacles, I think we've all learnt the importance of tenacity and perseverance in trying to roll out change. And I think sometimes people tend to give up because it's too hard, but I think if you keep going, eventually you get your message out there, and hopefully we'll get this rolled out.